Well, our last speaker before the lunch break is Dr. Babu Valliyodan. He's a senior research scientist from the National Center for Soybean Biotechnology and Division of Plant Sciences at the University of Missouri. Dr. Valliyodan received his PhD in plant biology from India and studied molecular biology of abiotic stress tolerance at the National Research Center on Plant Biotechnology, Indian Agricultural Research Institute in New Delhi. After his initial training, he joined the Soybean Genetics and Genomics Lab at the University of Missouri and started working on the research projects focusing on trait discovery associated with abiotic stress tolerance and yield improvement in soybean. Right now, he's focusing on, or he's actually leading, the soybean resequencing project at the Soybean Genetics and Genomics Lab at the University of Missouri, along with the project director, Dr. Henry Wan. So please join me in welcoming Dr. Valliyodan, and he will be talking about breeding climate-smart soybeans.
Okay, before going to my talk, I may give an introduction about our center. National Soybean Center. We established the center about 12 years back from the support from United States, USDA funding, and the University of Missouri Kaffner. So we are about 40 scientists working on various aspects of soybean soybean biology to improve soybeans. Our mission is to provide solutions to soybean improvement in the United States and provide educational and industry and international collaborations. So as you can see from our center's theme, one of the major themes in our center is the abiotic stress tolerance, mainly drought, temperature, high temperature, and flooding stress. So we are about 15 or close to 15 scientists working on the various aspects of the abiotic stress conditions in the center. So while I'm looking for, actually I was preparing slides for this talk, so I found this interesting slide from the ICRISAT website. Indeed, we are living in the midst of a perfect storm of all these major challenges in the human life, especially from the people from the developing countries. So the number one challenge is the climate change. So climate change, we all know that it threats our ecosystem, food, water, and economy. And also, we know that by 2015, the population explosion requires about double the agricultural production. But due to all these challenges, it's very difficult to achieve that point unless otherwise we design our cropping system or design our genetics or plant genetics to achieve that point. This just show that this is a slide I got from World Bank. They project about in 2015. What will be the status of a crop production? These are the hot spots where the crop production goes to below 20%. So from those slides, we can see that major challenges are the rising temperature, drought, flooding, and weather extremes as per the IPCC meetings. So when we look at the major crops in the United States, the recent drought in last 25 years affected most corn, soybean, and wheat production in the country. And you can see that about 40% of the share is from the extreme drought conditions. So from this extreme drought conditions will affect definitely the income, farmer income, or the state income. Last year, or a couple of years back, it's about 25 million loss only in Missouri state. mainly on soybean and corn. So as we all know that, soybean is one of the major agricultural product, or the legume crop for human, animal, and fiber. And we are the #1 producers of soybean, and we can look at the world production. This map shows that major agricultural production areas in the world where the drought occurs periodically, or the drought, extreme or historic drought there. But interestingly, we can see that major soybean producing countries fall in this map. So drought and high temperature are the major factor limiting the soybean production in the world. And you can see that even though the graph goes a little bit up, but the new technologies are needed to meet the graph go up. That means to keep the production system going up, facing these challenges like drought and high temperature. So by looking at the challenge, the environment challenges, this is a kind of a quote from one of the program officers at the Gates Foundation. Says that the best adaptation to climate change is breeding and seed system that rapidly develop, deploys, and replaces varieties to farmers. So all these introduction slides shows that, yeah, we have to look at these challenges and dissect those specific complex traits like drought and temperature influence on crop production. So we use a kind of integrated approach for this, for dissecting this crop production challenges. We use we conduct all the phenotypic analysis. We screen soybean germplasm and select the soybean genetic resources and use those genetic resources for dissecting genetic variability, either by classical genetics or the next generation genetics recently developed using various tools like omics tools or genome sequencing tools. Then we identify genes or QTLs, either use the forward genetics or the reverse genetics approach to get the soybeans with a better drought tolerance or better stress tolerance and improved yield. So what we got, for soybean, we have a complete genome sequenced in 2010, and we have the new version of the genome came recently in December, November last year. And also we have about 20,000 soybean accessions or germplasm in the United States USDA germplasm center. Out of this 18 or 20,000, about a couple of thousands are kind of wild soybeans collected from various parts of the world. So that's one of the major resource for us to go further. So our approach is, as I mentioned before, our approach is to look at the not only the phenotypic screening, we look at the genotypic screening, like a genome, how the genomes are different, and what are the genomic signatures responsible for this drought responses or high yielding or whatever it is. So for that, we have a huge project on genome resequencing, and we selected about 1,000 soybean genomes for resequencing to identify these signatures associated with these stress tolerances. So our major goals are sequence those genes, the cultivated soybean plus some of the wild soybeans, then make more reference genome. At present, we have only one soybean genome, that's a Williams 82 variety, and collaborate with the international community like China, Argentina, Brazil, and other major soybean production countries to get the information and get the more genomic information to find out or to investigate desirable traits. So at present we are sequencing cultivar Lee. That's kind of mainly on the southern maturity group 4. The one we are available now is the maturity group 3. That's Williams 82. So we are making a genome of Lee, and also we already finished about 500 soybeans sequenced, and more analysis is going on. The second phase is initiating. It will be initiated in February to sequence the next 500. So these are the resources what we have it now. We have a genome completely sequenced. We have about 20,000 accessions, including wild, and we have close to 1,000 genome sequences. Plus some of the ancestor lines from other like Iowa and Maryland USDA sequences. So utilizing this genomic resources, we look at the drought resistance mechanisms, mainly on drought avoidances. So I'll be giving some highlights of the research what going on in our group regarding drought avoidance. The major drought avoidance traits are kind of root-related traits or water use efficiency traits, transpiration efficiency traits, and I'll be giving a couple of examples on the slow canopy wilting and water use efficiency traits. Due to some of the IP issues, I couldn't put more data over here. So this is a famous equation. Yield, the better yield, you should have a better water capture, water use efficiency, and harvest index. So you have to look at an integrated system, both root and shoot system, to achieve the better yield. So for the drought avoidance, we select genotypes or the soybean accessions from the germplasm bank suitable for the Missouri growing condition, mainly maturity group 3, 4, and 5, and look at the slow wilting under reduced soil water and yield loss and the greater root growth under limited water conditions. Why? Because it's a very complex trait, and it's very difficult breeding this trait. And up to recently, we didn't have much information on this traits. And recently, we found a couple of traits like slow wilting are important in the addressing this drought and the high temperature issues in soybean. Slow wilting issue, slow wilting or canopy wilting issues are addressed recently from a group from North Carolina, Tommy Carter and people, and identified that these traits contribute mostly towards a better yield under stress conditions. And we have a couple of projects on this funded by the United Soybean Board. So what we did, we selected about 800 or close to 1,000 soybean lines from this maturity group, 3, 4, and 5, and screened under both greenhouse conditions, grow chamber conditions, and field conditions, and using the better phenotypic screening strategies, like 3-tier strategy. First, you do a kind of a high throughput, then you narrow down, a funnel down, and select some lines and screen it for several years, like 5 to 6 years, to identify the lines. So this photograph shows a couple of the lines going at the Clarkton farm. I mean, no water for about 6 weeks. And from this screening, we can identify sensitive types and tolerant type soybeans. And also, we can look at the yield or harvest index by comparing the sensitive and the tolerant soybean lines. So what we got, at the initial screening, we got about 36 3, 82 4s, and 18 5s, very promising lines, and we continued growing this in about 5 to 6 climatic conditions or growing regions like Kansas, a couple of places in Kansas, and in Portageville, Missouri, and Clarkton farm. So after all this screening for the last 5-6 years of this 1,000 lines, and narrowed down to close to 11 soybean lines with a better drought tolerance and a better yield. So we found that some of the lines segregate both for slow canopy wilting and a better root system characteristics. So after getting these PIs, what we do, we look both physiology and the genetics or genomes, and also make some genetic investigations using this PI. So at first we did some physiological investigations, and we found a couple of the gene families, such as aquaporins, are regulating or controlling this canopy wilting in soybeans. And we did a rigorous RNA or transcript analysis of these particular PIs using modern transcriptomics tools, identified these specific candidates. And we are testing these candidates in the model systems, and also we are looking to develop markers or functional markers for those candidates. Another example is that we found some other organelles, like plastoglobules, associated with the drought tolerance. It's mainly associated with the chloroplast. So this may be one of the reasons regulating the canopy wilting in soybean. So coming to the genetic investigation, we use this soybean lines and make crosses and develop mapping populations. And the mapping population segregates in a different way. You can see that it segregates for leaf shape, and also it segregates for other characteristics like lodging. And the recombinant lines, the recombinant imprint lines, segregate for early maturity or late maturity types. So what I mean is that even though we use this kind of materials, it need rigorous selection or a rigorous screening to identify the line or soybean line with a desirable trait in, then use those lines to incorporate into the elite lines used in the various climatic conditions or growing conditions. So next, the root traits, why the roots are important, and one of the speakers talking about the deeper root system for better survival in other crops. Yeah, the roots are the number one organ in the plant, sense this water scarcity well in the land. And we can see different phenotypes of the root system associated with stress tolerance. So, like in our shoot system, we selected about the same number of lines, maturity group 3, 4, 5, plus some of the wild and perennial soybeans. We screened for the root system architecture. So you can see the different root system architecture in different PI or plant introduction lines, and also some of the potential drought tolerant lines. And then based on this root system architecture, we selected some lines to make a genetic population, a mapping population, and also to study the genomic or the physiological mechanisms associated with the root trace. Either it's a root angle or the deeper root growth or more lateral root, or and so and so. So at present, we don't have much QTLs identified or genomic regions identified associated with root traits. And recently, we have identified some. So another thing is that we use both cultivated and wild species to get the maximum diversity to get maximum diversity and maximum rooting capacity in the plants. So one of the mapping population is we used a Dunbar and a Soja population. Then you can see the difference in the root system architecture under different time of growth. So these are the parents, the Gmax and the G-Soja root system architecture difference, and after the recombinant inbred line have a different root system architecture. And like in our slow wilting characteristics, we identified soybean lines, or identified the QTLs, or genomic regions associated with taproot length, lateral root number, and the shoot length using this population. And we are investigating, we are mining this QTL region to identify the specific candidates for next generation breeding or next generation engineering of soybean. Another population we used is a kind of is also a kind of a Vmax, Gmax, and a soja population, and the recombinant inbred lines, the segregates for root system architecture. Some of the examples I have given here. So we found a couple of QTLs in chromosome 6 and 7 associated with root system architecture, like root angle or deeper root growth, and we got more than 12 to 15 centimeter in some of the recombinant inbred line, the root length, taproot length. And so after that initial QTL studies, we sequenced those parents and also some of the recombinant inbred lines to go a little bit deep into the QTL region. Then we identified a couple of the genes associated with that trait. This is another example of, first is chromosome 6 and some of the chromosome 7. So I forgot to mention that we are using a new technology or kind of a newer technology for looking at this kind of traits. We use kind of genotyping by sequencing. So that means we sequence the parents very in a deep, kind of a 15x to 20x genome equivalent. Then we sequence all the 500 or 400 recombinant inbred lines to kind of a 0.2x to 0.5x genome equivalent genome sequence. Then use those genome sequence to identify genes or a genomic signature associated with a specific trait. Indeed, we need a high throughput phenotyping platform for all these studies. And thanks to MU Kaffner and the funding to the Bradford firm, we have a better rainout shelter over there. Felix and Bob Sharp and team established that funding to get that rainout shelter for our drought screening. And also we have developed a different greenhouse and field screening for root system architecture. So along with this screening, we use a new genome-wide association studies. That means we selected lines from different climatic conditions and different areas. Then we sequence them and look at the genome sequence and identify specific QTLs associated with the particular trait. We call the genome-wide association studies. Then use those information and for better gene discovery or the marker discovery for applying in the molecular breeding. So in a nutshell, genomics and the physiological investigation provides several candidates, and we have a lot of candidates available now, and we are developing markers. And we are using those genes or the markers or the genes to incorporate into elite lines that for various climatic conditions can be grown in different areas of the country or different areas of the world. And this is the flowchart we normally follow for the development of the new varieties. And at the same time, we look at these genes in the reverse genetics too. So we look at the functions of the gene. Earlier we looked at in a single gene by gene, but now we are looking at kind of a gene stacking pattern. So like we put about 4 or 5 or 6 genes together. There's a couple of examples that show that with the different components like drought avoidance component or heat tolerance component or yield improvement component, put it together and test those genes in soybean. And some of them are coming up. So some of the greenhouse experiments that we found, one of the gene construct shows kind of a little bit better survival in the greenhouse condition, but that won't guarantee that it will translate better in the field condition. When we look at the field, yeah, we're still kind of growing those transgenics in the field condition. Still, it's preliminary. We found a couple of green signals like improved biomass and some of the pod number changes in some of the lines, and likewise another candidate gene we transformed also shows better biomass and the field conditions. So I gave a kind of a flair of what we're doing there, and we do mainly a lot of forward genetics approach using this genomes and germplasm resources. And at the same time, we use reverse genetics also to test some of the candidate genes and to take it to the field. So in summary, we have contrasting soybeans with a contrasting root system or canopy wilting traits. We have genes and QTLs, and we have markers, and we are developing strategies for the next generation breeding and the gene stacking technologies about multiple genes to get our trait for better yield. So what the future? Yeah, like all other crops, like we should get more yield, and we need plants to adapt to various environmental conditions. Plus, we have to develop a breeder's toolbox, user-friendly breeder's toolbox mainly for the soybean breeders, that they can use all these technologies, the new genomic technologies. Like you can take a small sequencer to the field, and if you find a line, so you can sequence the field itself within 20 minutes. And the new technology called complete genomics offers that kind of technology. Then you can process the data quick. So now from the 20 billion genomes to kind of a $1,000 genome or $800 genome now. So it's easy to identify the signatures for the breeders. They can use that as in the toolbox. So this is a huge project, and a lot of people involved in this aspect of the project. And from MU, Henry PI, Silver's mainly working on the root, Raymond on the shoot system, Song and Lee working on the reverse genetics aspects, Dong, Tripty, and mainly on the bioinformatics side. And we have breeders like Andrew Grover and Transformation Zhang. From North Carolina, Tommy, and from Danforth, Charles Ann, and from the breeders from Arkansas. And because we test all these lines, all the PIs over these different areas, growing conditions, and this is a huge collaborative project funded by mainly this project funded by the United Soybean Board and WARA and Nebraska Jim, and we have an international collaboration set up recently mainly on the genome sequence. Then in the exchange of information from China, the National Center for Soybean China, and in Korea, National Center and ICRISAT, and from Brazil. The funding support from USDA, United Soybean Board, and Missouri Soybeans, and these are our collaborators, BGI, Beijing Genome Institute for Sequencing, SoyKB, all the data what we produce, all the sequencing, all the marker, all the QTL data, everything you can get it from the SoyKB. It's hosted from Dong Shu's group, so you can go there and get all those. Thank you very much, and ready for questions.
All right, thank you, Babu. We do have time for questions, at least a couple of them.
Very interesting work. Where do you see these drought-resistant soybeans fitting into an agroecological cropping strategy that can increase the land equivalent ratio that Tom Salyer spoke about in the keynote?
You know what? I mean, the forward genetic approach the major goal of this approach is to get new varieties. So one of the approaches in the climate-smart agriculture is to provide new varieties to utilize the available land. And definitely, I'm sure that we are very close to that. And we have a couple of varieties coming up, a couple of the lines coming up. So hopefully, it can be used. And recently, some of our colleagues are involved with a project, USAID project, focusing mainly on some of the African countries Kristen Bellew, Felix, and Henry. So they are trying to do some kind of a breeding, sort of a breeding wing over there for the small-scale farmers, to help the small-scale farmers over that part of the world. So yeah, we definitely translate whatever we get in our labs to this kind of approaches. Hopefully I addressed your question. No?
You focused on drought tolerance development in these varieties, but as Dr. Sora pointed out earlier, climate change also brings extreme variability. Here in Missouri in the last 10 years, we've had 2 very hot years, but 3 of the wettest years on record as well. So I'm wondering how your drought-resistant varieties respond to extreme rainfall events, whether singly or collectively, given that that's been just as common in the last decade as extreme drought.
You know what? That's a good question. So recently we started working on the interaction between drought and heat stress. So we are kind of screening more germplasm in that with that aspects, like using different heat tents in Kansas. So at present we don't have much answers to that point.
On your work is going to basically go to breeders, as I understand it. So will this information become more public access, or will these seeds be eventually patented and made private like the current Germplasm that we're dealing with now?
So most of this kind of, since some of the nature of the funding, like some of the fundings from the United Soybean Board, and most of these findings goes for the public access, definitely go to public access. And at the same time, some. Yeah. So that depends the funding though, and some of the findings from the United Soybean Board projects. So that definitely goes towards the public access, and some of the Missouri soybeans, some goes. Yeah, it goes to public after Missouri soybean farmers, IP issues taken care
of. How transferable is the methodology of sequencing and measuring of the genes to other crops, other vegetable crops or grain crops or perennial crops such as nut trees and oil trees?
It's very much, and like you can see that this started in the model species, like Arabidopsis earlier, like about 5 years back. They sequenced about 1,000 to 2,000 Arabidopsis lines. Now, lot of crops like rice. Recently, they published about 5,000, and maize, it's about close to about 1,500, and tomatoes, and lettuce, watermelon. Yeah, a lot of crops are kind of now they're using this genome sequence-based breeding design or that kind of approach. Yeah, it's kind of, nowadays this sequencing cost is very, very less. I mean, it's not like a $2 billion or a $20 million now. It's $500 or $1,000 you can get it.
Just for clarification, when you're talking about genome sequencing, transgenic, this is all referring to what maybe the average consumer knows as genetic modification. Is that correct?
I mean, genetic modification. That you know what? That terminology can be defined in a different way. You know, even in the case of the classical breeding, it's genetic modification, right? It kind of by genome sequencing, what do we get? We can see what is in the genome. What makes this plant, and what makes this more leaf or less leaf, more drought tolerant. So if you can find out those specific hotspots, you can use that either for the classic, the breeding approaches. Like you can incorporate those hotspots in your the elite cultivars or the lines growing in your area of climatic conditions by so-called molecular breeding approaches. So at the same time, we can use those information, or we can validate those information, or use the information to engineer those genes, the same plant gene in the same plant, not from any other, same plant in same plant, to see whether we can get the phenotype or not. So we can use both, like both forward genetics and reverse genetics utilization of these genomes.
So I know in Europe there's been a lot of studies done on the human health effects of genetically engineered crops. Has any of that been done here in the U.S. with regards to these genetically engineered soybeans?
I think that a lot of work's done, and I don't think that kind of. I mean, always it's there. Therefore, I mean, I don't want to go into that debate. You know that 90% of the soybeans in the United States is genetically modified, and because of the Roundup Ready and all other thing. But as a soybean, I mean, as a scientist working in soybean, I didn't see any negative. I can put it that way. A negative impact of those genetically modified soybeans in humans or an animal feed in the United States. That's my personal observations. Yeah, definitely. We all scientists, the academician in the academy, we definitely have a moral obligation to give better product to the community. So we should look at it in a very serious way.
One more question.
Yeah.
I'm really interested by the potential for transgenic research to provide sort of genetic sanctuaries for the wild varieties of our food crops. So where were you pulling these wild glycine from, and what is the conservation status of those wild landscapes?
You know what? I don't have much a good answer for that, but I can say that most of the wild, like about 2,000 or 1,500 wild soybeans, we collected from the various parts of the China, Korea, a couple of Japan, some from India, some from Russia, and a couple from Norway. So these are kind of, in some places, like when I talked with a collaborator in China, we found that they have a program to conserve those lines what they have. In Korea also, the group called Dr. Lee, they are trying to conserve that. Other than that, I don't have a good answer for that thing, and we have that resource available in the gene bank, and we use it. And I mean, one thing I can tell that these are kind of an untapped resource. These wild soybeans or perennial soybeans are untapped resource for most of the agronomic or quality traits. So, I'm sure that it will be the same with the other crops. Like recently, a publication in sorghum. They published in Nature Communication. They found that some of the wild sorghum genes are how much it is important to the sorghum quality, and watermelon also.
Okay, the last question.
Going back to the GMO crops, when we talk about BT, then that's an introduced gene from another organism. But when you impart drought resistance, which is from the same crop but just a different cultivar, do you think those are safer compared to the BT introduction of BT genes, which is a different organism itself?
Yeah, that's a tricky question. So you want me to claim that the BT transgenics are bad? No, I don't make any comment on that. So we are using, for the transformation, it's very safe. Not very safe. It's safe. I can put it that way. That we are using the same gene from the same plant, and putting in the different varieties, like either Williams 82 or Clark or Jack or some of the PI lines what we just mentioned about it. So
to follow up, so that means that gene was existing in the same plant naturally. So my question is, it should not be any harmful, even if it's genetically modified organism.
Possibly not. Yeah.
Thank you.
I was a little bit reluctant to put the couple of slides, the last slides for the transgenics, because I don't want to get any debates on this in this audience. But I want to show that, we have both the reverse and forward genetics approach in the crops going on in our group.